Hello, everyone, and welcome to phyloseminar.org. The current theme is machine learning for the second time, and this is the fourth talk in a series of now 5 talks on that topic. Please use the YouTube live comment box to ask questions. Today's speakers are Ammon Thompson and Michael Landis. In 2015, Ammon earned his PhD in evolutionary biology at the University of Texas in Austin, where he studied the molecular evolution of gene families. He then worked as a postdoctoral fellow at the University of California, Davis, where he developed statistical models for analyzing organ transcriptome evolution. Currently, Ammon is a participant is a scholar at the Oak Ridge Institute for Science and Education. Michael received his PhD in integrative biology from UC Berkeley, studying with John Holzenbeck. He then did a brief postdoc with Tracy Heath at Iowa State University, followed by another postdoc with Michael Donahue at Yale University. He's generally interested in phylogenetics and in particular models and methods for investigating the evolution, ecology, and biogeography of species. Michael is currently an assistant professor in biology at computer science and engineering at Washington University in St. Louis. Welcome both, and thank you for participating.
All right, all set?
Take it away, Michael.
Okay, well, thanks, Eric, for the introduction. Like you said, my name is Michael Landis, and I'm here today with Ammon Thompson. And we're going to share some new analyses we've been doing and studies we pursued concerning how deep learning can be applied to, in this case, viral phylogeography, but potentially to a broader class of phylogenetic models. The structure of this talk is a little bit unusual because we have 2 speakers. It'll look something like this, where I'll introduce a little bit of ideas that we have on how do we actually design phylogenetic models and how do we use them. Then Ammon's going to describe in more detail exactly what we were doing regarding deep learning applied to viral phylogeography. And then I'll return at the end to talk about how might we design deep learning pipelines for studying phylogenetic models in general. And I would say this is sort of a sandwich format where really what I'm going to be talking about are fairly conceptual, somewhat fluffy compared to what Ammon will discuss, which is the real substance of the talk. Okay. Now, just for broader context, this project that we'll be sharing began in about 2021 of spring. And as many of you will recall, COVID was still in full swing. Many people were isolating, baking bread, things like that. Around that time, Ammon and I began our collaboration thinking about how we can model phylogeographic processes applied to viral evolution. At that time, we also had some really cool new tools popping up in the popular in a popular setting, such as DALL-E, which is this tool was one where you could enter a phrase and it would render a series of images that basically correlate with that phrase using deep learning approaches, and it's really kind of magical. You could conjure avocado chairs from thin air. Around that time, as Ammon and I were collaborating, we also read this really cool preprint by Vosnica and others, and I believe they spoke 1 or 2 sessions ago. So check that phyloseminar out, where they introduced a phylogenetic deep learning framework called PhyloDeep. And the key technology they used were these convolutional neural networks. And these are very similar to the types of neural networks that are used to generate avocado chairs. So, needless to say, Ammon and I were intrigued and learned a little bit more. Now, in terms of the modeling setting that we'll be working on, it's basically a viral phylogeographic setting. Viral phylogeographic analyses tend to look something like this. They tend to be organized around having a time-calibrated phylogeny. The taxa in that phylogeny are serially sampled over space and time, meaning that we're going out into populations, sequencing infected individuals, and recovering the viruses that they might be infected with. Then using that information to reconstruct phylogenies. And with these types of models, you can ask lots of questions that are interesting to evolutionary biologists, but also public health officials, like, do viruses move from region 1 into region 2 faster, say through interregional transmission? Do viruses replicate faster in region 1 or region 2? And do viruses seem to be more prevalent or is sampled at a higher rate in region 1 or region 2? Now, in terms of the anatomy of these models, many of these models generally have these types of events underpinning how you generate data patterns. They tend to have events that will be familiar to you if you work in phylogenetics as a whole, such as infection events or transmission from an infectious host to a susceptible one, or a birth event, a lineage splitting event, migration or transitional events between populations, such as these population types 1, 2, 3, 4 at the top, from black to blue in this case. You also will have host recovery. The host develops immunity to the infection, or perhaps they die, and the virus can no longer replicate within that host. and sampling or detection. When do you actually get the sequences from these different hosts among populations? Now, these types of models, I'd say, fall into a fairly general class of models. They're all analogous and even equivalent under certain conditions. And they appear with different names, such as state-dependent speciation extinction models, or SSE models, multi-type birth-death models, and even in some cases, you can couch these as susceptible infectious recovered, or SIR models, with a migration parameter. Now, the reason that we like these models is, one reason at least, is that you can estimate the parameters that generate the data set. And what's cool about that is when you get these estimates, you can potentially use the equalities or inequalities of those rates to do hypothesis testing, such as the ones that I presented before in the example with Sierra Leone. So do transition rates between regions differ? Which is a source region, which is a sink region for the spread of the virus? Do infection rates differ among regions, and so forth? And here are the types of verbal hypotheses that would correspond to these types of equalities and inequalities you might estimate. Now, one complication is that on the left-hand side, I've shown you what might be called the complete tree, which is, you could consider this as a fully observed sequence of evolutionary events that generate the data set. In reality, the problem is that we have an incomplete tree with imperfect sampling. And on top of that, we don't know the timing, type, or number of transition events between, say, populations in this case. Now, let's take this a step further. Suppose what you're interested in is not just estimating these rates, but also learning what are the things that govern these rates, such as what might be governing the rate of viral transmission between species. So this is a figure from this Wells et al. Paper where they're looking at different reservoirs for SARS-like coronaviruses, which are known to be found in bats, pangolins, and civet species. And at least in multiple times, they appeared to have transitioned from those reservoir species directly into humans. So if you start thinking about what factors might allow these interspecific viral transmissions to occur, let's focus on ecological factors for a moment. By and large, if you're going to have transmission between species, those species probably have to co-occur in space. When you start thinking about the biogeography of species, namely the hosts and viruses that are involved, what can actually allow for sympatry or co-occurrence of species? Obviously there's a geographical component to it. What is the species range? But there are also many important environmental and ecological factors, such as what ecologists use to represent, say, the niche of a species, where they can occupy in climatological space. What is the actual ecology or ecological niche of the new host relative to the current hosts that are that can be infected by that virus? And this paper here by Gavat and Frutos named, say, 6 relatively important parameters that might govern these types of interactions. And they didn't stop there. There are other types of factors that could potentially influence the rate at which viruses switch or expand their host ranges. Not just the ecology, but what about the ability for a virus to evade the immune system of the host, to hijack their cellular machinery, and so forth? And what about the capability of the virus to basically exploit host behavior and allow that virus to spread among individuals in the population? There are lots of different features that might contribute to how, when, where, and why these viruses are transmitted between species. So it's a pretty complicated modeling problem when you get into the details. Now, I mentioned that these diversification models that might be used for viral phylogeography can be couched in different ways, but one of them is this SSE framework. And what's wonderful about the SSE framework is we can get a likelihood out of the model. It's a pretty complicated model. However, the framework that we typically rely on to do estimation for SSE models is a likelihood-based framework. And what's really unfortunate is that the framework we use to compute those likelihoods is not an exact closed-form likelihood equation. We typically rely on these numerical ODE solvers to approximate the likelihoods. And they approximate them quite well. The downside is that they don't seem to scale very well, especially with respect to increased number of states. They scale reasonably well with increased numbers of taxa. But as you add model complexity, this machinery will break down. And just to step back for a second, I come from this Bayesian and likelihood-based background. In grad school, I was sort of, it's nobody's fault exactly, but I was sort of steered away from machine learning and likelihood-free approaches. And I was always taught, to do inference properly, you have to have the likelihood. And, during the pandemic, I guess I was looking for easier ways to do things in general. And one morning, my kids, early in the morning, I should say, my kids wanted waffles for breakfast. And lo and behold, I was out of batter. And I certainly wasn't going to drive to the store to pick up the ingredients. And so, trying to think quickly and maybe desperately, I threw a piece of toast into a waffle iron. And it turns out they were perfectly satisfied with this little waffle that popped out of the, this waffle that popped out of the iron. What it turns out they wanted was something starchy with squares that they could pour syrup into. And, I have to apologize because most of my anecdotes are about my kids, and this might be far reaching, but there's something interesting about this when you think about likelihoods. What is the purpose of the likelihood for estimation tasks? It's to get the parameter estimates. It's sort of the thing that we're using as a vehicle to get the actual hypothesis testing framework going. It's not true that in all cases, all biologists want likelihood functions. They just want the parameter estimates in many cases, right? So, thinking about likelihood functions and waffles and whatnot, it's good to at least pause every so often and ponder, what types of models would we be exploring as phylogenetic biologists and evolutionary biologists more broadly? What models would we be exploring? What would we be designing? How would we be using these things? If likelihood functions were free and inference was trivial, I think we'd be exploring a pretty different set of models than we explore currently. And these model features that I highlighted for SSE models are not the only features that can break our standard likelihood computational machinery, the stuff that is typically built on the back of Felsenstein's pruning algorithm. There are many biologically realistic modeling features that are not really compatible with simpler computational algorithms. Things with large and unusual state spaces, complex types of rate variation, trees within trees or reticulation, and so on and so forth. So this is just to set the stage for what Ammon's about to present here. He is going to do an assessment for how these deep learning methods, such as the ones introduced by Vosnik and others, and our extensions to that approach, how might those approaches compare to the gold standard likelihood-based approaches that we do trust in the field? And as a test case, he's going to take these viral phylogeographic models, which do have a handful of features that make them not fully extensible for likelihood-based inference. So we're trying to find a breaking point. When does deep learning fail? And I was sort of surprised by these results. I think you'll be interested to see what Ammon has to share here in a moment. And once he's done, we'll come back to some other future directions that we're working on currently. So let's swap over to him now.
Thank you very much, Michael. Yeah, that was a great introduction. And as a parent myself, I appreciate the waffle. Ammon, can you bring your slides up? Perfect.
Okay.
Great.
Thanks. Yeah, that was a great intro. So for my part of the talk, just a quick outline. I'm going to give a little more background on the challenges of using phylogenetics for epidemiological inference. And then I'll talk about some recent developments in using deep learning to overcome these challenges. And then I'll tell you about some interesting experiments we ran and how we applied deep learning for inference about viral phylogeography from trees and location data. And some experiments that we ran to compare the robustness of the two approaches, a likelihood-free deep learning approach and a typical likelihood-based approach to inference. And so in the context of epidemiology, usually by the time an outbreak is detected, a virus has usually spread some distance from its epicenter. And so one of the reasons epidemiologists are so interested in phylogenetics is it's a way of taking, more and more plentiful genetic data from viruses and filling in some of those knowledge gaps about what happened in the past and inferring a subsample of the underlying transmission tree that isn't directly observed. And so you can learn a lot from fitting epidemiological models to phylogenetic trees. And so that's typically done in a likelihood based framework, where we have tools for calculating a likelihood of a tree. So the basic epidemiological model is what's called an SIR model. It's a compartmental model where you take a population of size n and subdivide it into 3 compartments. One compartment is the subpopulation that is susceptible to infection. Another subpopulation are the infectious individuals, and then denoted by R are the individuals who have been infected but are recovered and are no longer susceptible. So the rates at which infections occur are density dependent. They depend on the relative numbers of susceptible individuals in the population. And so as an outbreak progresses, that pool decreases. And so the rate, the branching rate in this process continuously declines, yielding such curves as we see on the right, where there's a wave of infections. Now, to compute a likelihood of a tree, we can approximate at least the early exponential phase of an outbreak using a simple birth-death model. And so by inferring the parameters of birth-death model, such as the birth rate and the death rate, or speciation rate or extinction rate, we can then compute interesting epidemiological parameters of interest, such as the beta, or the transmission intensity, or gamma, the recovery rate. And so one challenge with this is that epidemic models, especially these compartmental models, it's a very versatile modeling framework. And so as a result, there are many different compartmental models that can be quite complex, and therefore very difficult to compute likelihoods of trees. And one important complication is that lineages do not evolve independently. And so for this reason, there are a lot of epidemic models that are very realistic, or at least biologically plausible for the particular infectious agent that they were developed for, but they are too complex to be able to do inference using phylogenetic likelihood-based approaches. So recently, Jacob Woznicka and colleagues over in Europe sidestepped the likelihood function by using a simulation-trained deep learning approach. So for them, what they did is they simulated outbreaks and sampled trees from those simulations, and then trained a neural network with those trees to predict the simulating parameters, such as the R0, which is called the basic reproduction number. It's basically the expected number of individuals an infected individual will go on to infect, and parameters like that are of interest to epidemiologists. And they showed that they were able to get estimates that were as accurate as a likelihood-based method. And so this was pretty exciting because it suggests that maybe we could sidestep the lack of a likelihood function for some of these really complex compartmental models, and maybe do some inference under those. So Michael and I wanted to see if we could adapt this approach to also include location data so that we could do some geospatial epidemiology inference. And so with this approach, what we need is a way to simulate an outbreak that spreads among different locations and sample trees from that population of infectious individuals. And so a really good program for doing that is this package called Master that you can run in Beast 2. So with this package, it's very flexible. You can specify pretty much any compartmental model you can imagine, which is, a system of coupled differential equations. And it's got a really nice compact way of specifying. So you can, for example, specify an SIR model with migration among locations with just these 4 chemical reaction notation. So that's really nice. And you can simulate these populations. So you get population sizes through time, as well as sampled phylogenies. And so we use that as our way of generating training data. And so once you've got trees, a tree itself isn't exactly a great format for feeding into a neural network. So another nice innovation from the Voznica paper is they developed this way of encoding trees called a compact bijective lateralized vector encoding. So the way this encoding basically works is after latterizing the tree, you do an in-order tree traversal to populate a matrix that is of size n by 2, where n is the number of tips. And so the size of this matrix grows linearly with the number of tips. And another nice feature is that each row of the matrix corresponds to a tip. So it's easy to add other tip-associated data to the matrix. It's very straightforward. So, for example, we just added 3 more columns to the matrix to include 1 hot encodings of locations. And so our whole pipeline is we simulated outbreaks under a basic SIR model with migration among 5 locations. And we simulated about 100,000 trees from that process. Then reformatted them into the CBLV format, and then train a convolutional neural network to predict 3 rate parameters of interest. The R0, the basic reproduction number, delta, which is the sampling rate, and M, the migration rate. And I should also point out that we also fed into this neural network the recovery rate, which we assumed is known for this to make these other parameters identifiable. Okay, so what is a convolutional neural network and why use that for phylogenetic trees? So convolutional neural networks are really successful for analyzing images. So what you see here is what's called a filter or a kernel. And so in this case, it's a 4 by 4 by 3 kernel that slides along the this 2-dimensional plane, as well as another dimension for the color, the RGB color. And this filter or kernel is a set of weights that are multiplied by the RGB values here, and then summed to produce a single output for that patch of the image. And so as that slides along the full image, what's being constructed is a feature map of that. So this feature map is going to be you can have as many different features as you like. So if I pass 10 different filters over this image, then that feature map, along with having the same plane as the image, will also be 10-dimensional at each point or neuron in that layer. So to just give a simplified numerical example, here you have a flat image, grayscale, or actually binary here. You have a kernel here with weights that are 1, 2, 3, 4, 5, 6, 7, 8, and 9. And then you have an image patch of the same dimension. And you compute the element-wise product of the image patch and the kernel, and then sum up those values to get your output, or your feature value for that patch of the image. And so then you slide the kernel along to the next patch and fill out your feature map that way. And so in this way, what you get is the neural network will learn various features that will be useful for the inference task at hand, or the prediction task at hand. So for example, what you see here after the image are a series of lines at various orientations. So usually the earlier layers of the convolutional neural network are learning these basic features. And so then the feature map is basically a high-dimensional set of different features at each location in the image, which then goes on to the next layer, which then learns higher order features by doing another feature map of the previous feature map, and so on until you get to the final layers that are fully connected layers, and are thus processing the feature maps from the entire image. And so this, like images, a phylogenetic tree is structured data. So the order of these, the value of these rows contains information about the tree, as well as the numeric values. And so we can slide a one-dimensional convolutional layer or filter along the tips of the tree by using, for example, maybe a 4x6 filter or kernel. And in that way, you can create feature maps of the phylogenetic trees, and then train a neural network to predict these epidemiological parameters of interest. So you don't really need to look at the details of this neural network that we trained, but I just wanted to just show what it would generally look like. So here we have, 3 parallel streams of convolutional layers, as well as a fourth stream for unstructured data, such as maybe some other information that you might have about the sample, for example, the recovery rate. All of that, after passing through those layers, then gets concatenated and fed through a final series of densely connected layers until they predict the R0, the sampling rate, the migration rate. Okay, so what we first wanted to do is see if we could actually train a neural network to perform as well as a mean posterior estimate of these parameters. So we generated a test set of the idea is to generate a test set of trees under an epidemic model, and then analyze each tree using the trained neural network and an equivalent likelihood-based model. So for comparison, we used a Bayesian phylogenetic method under an equivalent birth-death sampling migration model. We did this in RevBayes with the TensorPhylo library developed by Mike May and Xavier Meyer over at Berkeley. And then we checked that the coverage to make sure that the birth-death sampling migration model implemented approximated the simulating model well. So what you see here are 7 different highest posterior density intervals, ranging from the highest 5% density up to the highest 95% density. And the steps you see are show the expected coverage. That is the proportion of the test set that falls within each of those intervals. And then the gray band is the predicted 95% predicted interval you would expect if the coverage was, if it had the true coverage given the sample size of 138 trees. Okay, so these look pretty good. So it gives us confidence that we're not handicapping the likelihood-based method. And so we went on to make our comparison for our 138 trees. And so this is the result for, these are scatter plots of our CNN predictions for the R0 parameter, plotted against the true values, shown up in blue, and then the mean posterior estimate of the R0 plotted against the true values for the Bayesian method. And so what you can see is that both methods do pretty well. They both have correlations at 0.98 and 0.99. And then they also correlate with each other really well with the correlation of 0.99. And so we do the same for all 3 parameters, the sample rate delta and the migration rate n. And we see both performing pretty similarly. But interestingly, if you look at the bottom row in orange, you'll notice that the 2 methods actually are making predictions that are more similar to each other than to the true value. You see that the correlations are 0.98 and 0.99, whereas it's more in the neighborhood of 0.93, 0.95 against the true value. So that's pretty interesting, maybe a little unexpected. But this result indicate to us that we trained a neural network that seems to be efficiently extracting information from data patterns that allow inference that is pretty accurate and maybe about as accurate as it can be. But what we really care about is how robust this approach is in the real world, where, as George Box famously said, all models are wrong, but some are useful. So we went out to test the robustness of the 2 approaches and by designing a series of experiments where we simulate some outbreaks under a more complex model than the trained neural network assumes and the likelihood assumes, and then analyze them with these 2 misspecified inference methods, and compared the results to see, if maybe the neural network is wildly sensitive to misspecification relative to the likelihood approach. So our first experiment was to misspecify the R0 model. So the inference model assumes R0 is the same among all 5 locations, whereas the test simulation model allowed that to vary among locations. So the results we see here is that both methods appear to be similarly sensitive and yield pretty similar estimates. If you look down at the bottom row in orange, despite, for example, the R0 and the migration rates being pretty sensitive, having much reduced correlations with the true values, they're still very highly correlated with each other for the 2 methods. So indicating that it appears that the machine learning approach isn't any more sensitive to model misspecification in this way than the likelihood approach, and seems to be converging on the same biases or predicting the same wrong answers. So that was a pretty unexpected and pretty interesting result. We did another experiment where in like manner, we allowed the sampling rate to vary among locations. And again, we see a similar type of pattern where the same parameters are sensitive to roughly the same degree, but are still highly correlated between the 2 methods. We did the same kind of experiment where the inference model assumes migration rates are the same among locations. For the test set, we allowed migration to vary among locations. Again, we see the same pattern with both methods converging on similarly wrong answers. Did another experiment where we ran outbreak simulations among 10 locations rather than 5, and then randomly grouped 6 of those locations. And again, similar patterns of sensitivity and converging on similarly biased and biased in the same direction inferences. Finally, we checked sensitivity to phylogenetic error, where we generated phylogenetic error using a realistic way in which we would expect error to enter into that process. And again, we see a similar type of pattern where both methods look pretty similar across the board. So yeah, overall, it appears that at least for these this finite set of experiments, the likelihood-free approach doesn't seem to be more sensitive to model misspecification. And so we could maybe expect that it could possibly not give much more wildly wrong answers than a likelihood-based approach when analyzing data in the real world. We similarly looked at another neural network we trained, which we trained to predict the spillover location from outbreaks among the 5 locations. So this, instead of using a mean squared error loss function, we use a softmax function, which can output a probability distribution over the 5 locations. And so you can get a probability of predicting the correct spillover location from that. And so analyzing under the true model, we see the distribution among the 138 trees, the probability of recovering the true outbreak location for the CNN, the convolutional neural network, shown up in blue, looks pretty similar, like a mirror image of the posterior mean, or the posterior probability of the outbreak or spillover location estimated using the Bayesian method. And when we look at all of the misspecified data sets and push them through our neural network, we do see that this holds across them as well, where you can see these two distributions of accuracies look pretty similar. So not just for these continuous variables like these rate parameters, but also for these categorical variables like the location of spillover, seem to behave similarly to the likelihood function in the face of model violation. We also wanted to explore uncertainty quantification with neural networks and how that behaves relative to credible intervals or the highest posterior density intervals from the Bayesian method. And so the goal for our neural network is to be able to predict intervals with approximately X% coverage. So be able to predict a 95% interval that has a 95% chance of containing the true value. And so the way that's done with neural networks is through conformal prediction. And a recent variation of that is a pretty nice method is called conformalized quantile regression. And so this method works in two steps. First step is to predict conditional quantiles. And then the second step is to estimate calibration terms to adjust those quantiles so that they will have the desired coverage or the accurate coverage. And so first, to train a neural network to predict quantiles, you just change the loss function to a pinball loss function. And so we just took the neural network we'd previously trained and swapped out the final dense layer and replaced it with two parallel streams of dense fully connected layers, where one produces an output for the lower quantile so that it has a loss function for the lower target quantile tau, and then another one for the another output layer for the upper quantile, which together will be predicted as a pair and can be used for an uncertainty interval. So the next step is to calibrate that. So this produces conditional quantiles. So, for small trees, you'll tend to get wider predicted intervals, and for big trees, you'll tend to get shorter ones. So in this example, the 95% intervals had a coverage of 79%, so too low. Both the lower and upper quantiles undercovered. So you can compute terms that you can then add to those quantiles to adjust them and shift them, either dilate them, shrink them, or shift them as the case may be, to produce a calibrated probability interval. And this one is guaranteed to have an approximately the correct coverage. And so we after training a quantile neural network, we simulated a calibration data set of another about 100,000 trees and used that to compute these adjustment terms. And then we simulated another test set to check to see if this actually worked and produced the coverages that they should. And so this is what we see for the three rate parameters. We check those same 7 intervals, quantile intervals. So for the inner 5% CPI, we observed coverages among the 5,000 trees of about 5%. And that was true, true across the board for all of these inner quantiles. So this indicates that it works pretty well. So we then went on to start comparing these results to their equivalent or their analogous highest posterior density intervals. So just to visualize a small subsample, this is what, a set of 25 trees, what the CPI interval from the neural network looks like in blue, plotted up near the highest posterior density interval. This is for the 95% intervals for the two, and the black dots are the true values. So you can see there's quite a bit of overlap between the two methods. When we compare the widths of the intervals, they're highly correlated with each other, which is good. And we see that the CPI, the neural network's widths, are about, on average, 20 to 50% wider than the highest posterior density intervals, indicating that this method, as implemented here, is a little less precise and more conservative than the Bayesian method, but seems to be a really nice first step in the right direction. So we then wanted to look at how sensitive are these intervals to model misspecification relative to the Bayesian HPDs. So here are the 7 intervals. This means we trained 7 different neural networks at different inner quantile levels and calibrated them. And so what you see here for the misspecified R naught data set, both methods show a pretty similar patterns of sensitivity to this model misspecification. And we see that among all 5 of the misspecified data sets, pretty similar responses for the two methods. So like with the prediction of the spillover location and the rate parameters, it seems like we're also getting our neural network likelihood-free approach doesn't seem to be wildly more sensitive than the likelihood-based approach to model misspecification. So our conclusions is that at least for this, these early sort of kicking the tires of the machine learning approach, it appears to be about as robust as a likelihood approach, which is nice because this machine learning approach is very flexible as far as specifying models. And it's very fast once you've trained the neural network, taking less than a second per tree. And these are like, largish trees. And we think, there's no reason not to expect this to be generalizable. And so I'm going to turn this over to Michael to talk about a cool Python package he's been working on to streamline this whole pipeline and to make it easier to specify models and simulate and train neural networks, as well as validate those approaches. So yeah, I'll turn it over to Michael now.
Okay. Yeah, thanks, Ammon. So I'm not sure how people in the audience will receive this, but I think it's fair to say that there's some potential for deep learning in phylogenetics. And if you're like me and you come from a likelihood background, then you probably are still wondering, okay, but under what conditions might deep learning be trustworthy for these types of estimates? Maybe you have different types of models. Maybe you've designed the network in different ways. Maybe you have different sizes of training data sets and tune your analysis in various manners. So how do you answer this broader question of establishing trust? Because I do think deep learning suffers from this problem that you don't have something that feels real, something like a nice function or that you can analyze and interpret to understand the performance. So, you know, in our lab with Ammon and others, we're interested in rapidly benchmarking deep learning under different models and methods to get a general sense of how these approaches might work. And, you know, programming these single-use pipelines is really not ideal. It's time-consuming. You end up with lots of incompatibilities across different networks and training data sets. And it really impedes the extent to which you can reproduce and share results. So what we're doing now is we're taking the experiences that we learned from Ammon's project and my experiences from working on RevBase for the past decade, and trying to find ways to find a middle ground in terms of how can we allow at least ourselves and hopefully some other people to use deep learning to explore modeling with phylogenetics. So the package we're working on here is called PHYDL, which is you know, PHY for phylogeny and DL for deep learning. And it's a pipeline-based toolkit for phylogenetic models and deep learning. The pipeline itself is true to its name. The steps are modular, meaning that the work for a step is done in that step alone. And the way that we pass information between the steps is typically using the file and project directories. So this is nice because it gives people an opportunity to take whichever steps of the pipeline they want to use and basically chop and screw it around for their own individual purposes. Now, I'm just going to step you quickly through a pipeline. I know we don't have very much time left at all, but the general framework looks like this. So pipeline is right now run just using a simple command line, though it also has a proper Python package on the back end. And so in this case, we're going to pass in a configuration file, you know, --cfg my project, and it will load a file below, that's represented below, which is essentially a Python dictionary. And let's start by setting up a model within PHYDL. So PHYDL will support a handful of built-in models, but you can also write your own Python class or use external model simulators. It's not a big deal. If you're using PHYDL for simulation, we're basically classifying models as having a general type, which defines the state space and the event patterns for that model type, such as a GSE model, a geographic state-dependent speciation extinction model. And then you can also assign different variants to models, and these will be, you know, things that essentially assign how parameters correspond to different event patterns, but also you can essentially augment the base model type with additional features. So later we'll be looking at a GSE variant with a density-dependent extinction event. So we've configured our model. Let's go ahead and put it into the simulating step to produce raw simulated data as output. So the simulating step, as you might guess, it just simulates large numbers of training data sets from the underlying model. And we have built-in parallelization support. I've been using it on my MacBook, but also on an Ubuntu server with 64 cores. Like Ammon had mentioned earlier, we're currently mainly supporting this master package, which is a Java software through Beast that lets you simulate phylogenies using these SIR models. And they're really cool and very flexible. One nice feature of them is it allows you to define evolutionary rates that depend on the states of other contemporaneous species. So you can have lineage interactions. After the simulation step, you essentially produce a large panel of training data sets, which include both simulated parameters. These might be the things you want to be your network to predict, the labels, but also you know, data patterns, input patterns, such as the phylogenetic tensors, or rather raw tree files and raw character matrices. I got ahead of myself. Because in the next step, we take those raw data sets and format them into this tensor format that most neural networks require as input. So Ammon had introduced this notion of the CBLV encoding and our CBLVS encoding. We have a few other encodings available through PHYDL, but essentially we are going to take the phylogenetic data and character data and turn it into a tensor so that we can train our networks. It's just a standardized format for processing phylogenetic state information. And right now we're using that with CNNs. In addition, the formatting step will take our training data and convert it into what we're calling an auxiliary data tensor, which will include summary statistics, you know, tree height, average branch length, and so forth, but also known parameters such as the recovery period for an SIR model. The other model parameters that generate the data set are put into the label tensor. These are the unknown things that you want to predict or estimate. So our training data here and our training labels here can be processed later on by the neural network. Once we have our tensors, we'll pass them into our learning step. And the goal of this is to produce a trained network that can make predictions on new data sets. Fiddle will produce a fairly stock network that you can use to input the tensors that it produces and generate point estimates and the lower and upper calibrated prediction intervals that Ammon had showed worked previously. And Fiddle will also do the standard things that you want to do in deep learning, such as shuffle and split your input into training, test, validation, and in our case, calibration data sets. And as part of this analysis during the training procedure, we also report in various ways how the training worked, both against the training data set and validation. Here we can see that the loss function for validation has flatlined, whereas it continues to decrease for the training data set, which is indicative, most likely, of overfitting. And so we probably don't want to train our network for that long. As part of learning, we also can compare the predictions under our training data set to predictions under a new batch of test data that the network has not seen before, and verify that parameter accuracy looks reasonably good, but also the coverage looks relatively well. And we report statistics for these types of things in the upper left. So next we have our trained network. We also might have a new data set that we'd like to make predictions against it and produce our estimated labels, our estimated parameters. So I need to slow down a second here and come up with a nasty model to work with. And the one that I settled on is a biogeographic one using the world famous Caribbean anoles. And this nice paper by Dan Roboski and Rich Glur in 2010, they studied a clade of over 100 anoles that are found in these 4 major island regions. And per island, you have about 33 anoles, 33 anole species. And based on their analyses with the models available at the time, they concluded that the slowdown in the accumulation of species over time is probably due to something such as competitive interactions or saturation of ecological niche space. Now, let's define a model with regional density-dependent extinction. In this model, when you have an island with just one species on it, that species has no competitors, so it only experiences a background extinction rate. What happens if there's another competitor? We are going to add an additional term for each competitor that species has to contend with. So now this species has the background extinction rate plus 2 additional extinction risks to contend with. And I'm a sort of a Mad Max fan, so for those of you who are familiar with the Thunderdome, this is sort of a 2 species enter, 1 species leaves type of process. Now, you don't need to understand the details of this, but I mentioned before that these SSE models, such as the BISI model, are a little bit difficult to work with. GSE models are also difficult to work with. Now, if a simple GSE model is difficult, a GSE model where you have lineage interactions is going to be even more difficult to obtain a likelihood function. And we're working on this problem with Albert Solongsono, who's a new postdoc in the lab, and also Ammon Thompson. And between the 3 of us, I think we're smart enough, and we have basically no idea how to get a likelihood function out of this. But fortunately, we have a nice deep learning pipeline that we can use to try to estimate model parameters. So we went ahead and trained our network using this model with perhaps 50,000 training examples simulated under master. And we estimated that a density-dependent extinction rate, which is, and with some back-of-the-envelope math, we were able to convert our density-dependent extinction rate into a carrying capacity. And this estimate would represent about a carrying capacity of about 38 anole species per island before we reach an equilibrium state of 0 net diversification. Now, keep in mind, like I said before, the mean number of anoles per island is 33. So it's not an insane result. And we did this without too much work, all things being told. So I wouldn't publish this, but it gives some promise for a type of model one might want to invest serious time in if you wanted to get a likelihood function. I wouldn't want to spend 5 years working on a likelihood function with no hope of actually getting a signal from data. So the last thing I'll touch on is this plotting step. You might have guessed by now, but all of the figures I've basically been showing for this section of the talk are generated automatically from using Fiddle based on how you set up the analysis. So, again, the main purpose of Fiddle is to let people rapidly implement, deploy, and inspect the performance of deep learning against different phylogenetic models. I'm really out of time here, so I have to skip through this. But we have we're hoping to release Fiddle sometime towards the end of summer. We have documentation. We have most of the core features in there. We're kind of in the beta testing phase at this point. So get in touch if this sounds interesting. The last thing I'll stress, because many people here are phylogenetic model developers, I tried to fairly quantify how much time I spent on this analysis. And we spent about 20 hours on model design, getting master to work with the GSE type of model and so forth, and some initial testing. For our actual simulated training data set, that took 6 hours on a 16-core MacBook Pro, which I'm speaking from today. The formatting step took about 15 minutes. Learning took 5 minutes. Prediction took a second. And the plotting was 15 seconds. So as a model developer, where do you want to spend your time? Guess what? Probably on the model design, not on all the downstream stuff. Now, if this is worthwhile, we should be investing time in developing likelihood methods, but I don't think we should always start with developing likelihood methods before we know if a model has any legs, right? I don't want to come across as being overly enthusiastic about this. I mean, I think like Ammon and probably many of you, I'm still pretty skeptical about what deep learning can do. There are definitely pros and cons to both a deep learning-based framework and a likelihood-based framework. I think with deep learning, some pros are that it's clearly fast. It seems to be scalable. It admits a lot of model flexibility. And if nothing else, it's something kind of fun and new to work on. In terms of cons, I do think deep learning often has higher hardware requirements, multi-core machines to do parallelized simulation. Deep learning does seem to be sensitive to various network, data set, model, et cetera, types of inputs, and that's not really well characterized. That's part of why we're designing Fiddle. Validating simulators, I think, is a little bit harder than we admit. And it either requires some clever math or another simulator implementation or some likelihoods to validate. Not entirely sure what to do there. And of course, a con is that in the same way that's new and fun, it's a little bit new and scary. I mean, it's not entirely clear how these frameworks should apply in all settings. So just to finish up here, I just want to leave with, if with nothing else, this question for you. What models would we really be designing and using as a field if likelihoods were free and if inference was trivial? Deep learning might at least push that envelope in some ways, right? It's nice for model exploration. I'm convinced of that. One thing I like about it is it doesn't only value the time of users, which is often a focus when doing method comparison. It really values the time of developers, because you can explore a model pretty quickly without needing to invest serious time in algorithmic work. And the last thing, for those of you like me who maybe were entrenched in their ways, just remember that not all estimates actually require waffles. Okay, so that's all. Again, I'd like to really thank Eric for running Phyloseminar for all these years and inviting us to speak. Ammon and Albert have been completely integral to anything that I've presented today. I am looking for PhD students and postdocs in general, so just get in touch if that sounds appealing. And we'd like to acknowledge NSF, WashU, ORISE, and Department of Defense for various forms of support. So thank you.
Phenomenal talk, both of you. Thank you very much. Very interesting. So I invite people to post questions. We are a little over time, but do you mind if we just go a little bit farther, another 10 minutes or so?
I have a few minutes if that's, if people are around, yeah.
Great.
And sorry we went over, I apologize.
No, well, we had that little, yeah, we started a little bit late. So this is going to sound like a trolley question, but it's not. So you are using the simulated trees, like the direct simulated trees. There's no phylogenetic inference happening for like the model training and the model validation. So the reason I hope this isn't a trolley question is I think that with usual likelihood-based Bayesian phylogenetics, there is a route to doing joint inference of the tree and the model parameters. And there are various people working on phylogenetics using deep learning, but I have not seen anyone attempt to do phylogenetics with deep learning with a really complex model. And so I'd be curious what your thoughts are about how we're going to sort of go in that direction.
Yeah, I have a few answers to that. Ammon, do you want to give your thoughts?
No, go ahead.
Yeah, I mean, so I agree that, in Bayesian phylogenetics, especially, there are lots of platforms out there that seem, they really are appropriate for doing joint estimation of phylogeny and various other types of model parameters. I mean, I work a lot in historical biogeography, and I'm really keen on how do you jointly estimate topology, divergence times, biogeographic history, dispersal rates, all of that stuff. And we're doing it in Rev-based primarily right now. I think with the deep learning piece, there are many labs, like you say, that are trying to figure out how to estimate, say, tree topology and divergence times using these neural networks. But trees are notoriously high-dimensional objects, and you seem to need to get a prediction that is high-dimensional to truly capture what that tree is. I'm not sure how people are going to do that, frankly, at this stage, using deep learning frameworks. There are two things I'd mention, though. Like, one thing is that when people are concerned about phylogenetic uncertainty, shall we say, one method is doing a joint estimation approach, and that properly accounts for all the say the distribution of trees that are compatible with the data that you have. That's great. Many phylogenetic biologists instead use these sort of serial approaches where you give it a distribution of trees and then make predictions against that distribution of trees or estimates against that distribution of trees and see whether that phylogenetic uncertainty has any chance of undermining the conclusions you want to draw based on the rate estimates. That's a great problem for deep learning, because I can go ahead and process 1,000 trees, each for a fraction of a second, once I have a trained network. So that's actually quite suitable. But the downside is it's kind of like it's a unidirectional approach. Like you have the distribution of phylogenies, and you don't let the downstream analysis actually reshape the distribution of phylogenies. So it's not great. The other minor thing I would say is I do think Bayesian phylogenetics is a great approach for joint inference for phylogenetic models, but it's only great for the phylogenetic models where we can get likelihood functions, of course. So if you start wondering about, well, maybe these models that we have are not adequate to really measure the things we care about, well, we still can't exactly use likelihood-based Bayesian approaches for more complex models. So in that sense, it might be a choose your poison situation for the time being. Hard to say.
Yeah, I don't know. Just it seems like you could. That was a great answer. It does seem like you could take your set of trees and do some sort of weighting or something like that. Like maybe have a neural network that takes lots of trees and for each one, maybe predicts its suitability for that model, and then does some sort of heuristic type averaging.
Yeah, almost like a neural network importance sampling kind of thing.
Exactly.
Yeah, that's cool.
Let's see. So there's a question that came in from Will DeWitt says, the misspecification robustness results are very interesting. He also asked, did you try misspecifications that violate lineage independence, e.g., could you introduce a carrying capacity in the test set? How robust are the deep learning versus likelihood inferences? And I think you had some lineage independence violations in your NL example, right? It wasn't strictly a carrying capacity, but...
In that case, it was like carrying capacity would be induced by the lineage interactions. So I didn't encode it as a carrying capacity parameter per se. And I guess I was just showing how the pipeline works. I haven't actually sat down and done serious analysis with that data set yet. So it would be cool to look at, again, how these more complex forms of model misspecification play out. But the issue there is we don't have likelihood functions for, GSE models with carrying capacity. So it's hard to, unlike Ammon's experiment, we don't have a good point of contrast necessarily. So it's not clear what to do.
Let's see. I guess, I mean, just another trolley sounding question. I mean, the kernel size does scale with the number of states, right? You had like 5 states and your kernel was 5 wide. Is that the approach that if we wanted to scale this to 60 states or something, would we try to, what would we do there?
Yeah, your kernel would have to cover all of those states if you want to get the structure of the, whatever your metadata is that's associated with the tips. There's a lot of information in the co-structure of where the tips are in the CDLV encoding and where the locations are or metadata is. So yeah, so if you took a kernel with, I've actually thought about, well, so what if you had your CDLV encoding and then you put, well, what if you did something with DNA sequence alignments or something, or you had some various haplotypes that are associated with some tips, and those contain a lot of sequence data. Yeah, it would be, I think, a good thing to explore sort of how does the time it takes to, say, train a neural network scale with that, and how many more data sets would you need to train a neural network if you've expanded that state space in that way. Yeah, I think it's one among very many different things to explore. I have a lot of robustness experiments that I wanted to do, but at some point I just need to stop and write a paper.
Yeah, no, it's really nice.
Yeah, there's sort of two ways that people deal with that. And in Fiddle right now, we have, there are three ways that people will deal with high dimensional data. And one way is kind of what we did, this one hot encoding approach. What's nice about it is it does not assume any linearity, let's say, among states that you're representing. And so you can basically really break any sort of artifactual correlations that might emerge from how you encode the data. So one hot encoding is nice for that property. Another thing you can do is just standard integer encoding. So with DNA, you might say that A is 0, C is 1, G is 2, T is 3. But in that case, you're implying an ordering that A's and C's are closer to each other than A's and T's. And that may or may not be a problem. Who knows? A third thing that people do, especially when processing text information, is you can start with the one hot encoding and then basically embed it in a different dimensionality. And the way that works, and I'm not an expert in this area by any means, but the way that it generally works is that you can basically find sort of axes that represent an axis that might represent a type of meaning, like a dog and a cat might be on a certain axis. And orthogonal to that might be lion and wolf, right? And so one might be about domestic versus wild, and another might be about type of animal. And so instead of having 4 distinct layers for one hot encoding, you can reduce it to just 2 axes. And so there's potential for that too. But what's tricky about that is, with semantics, I have an intuition for what that means. With evolutionary characters, it's not really clear like what those axes should be or how you define them. And people they basically determine what those axes for embeddings are through algorithmically. But I just don't have a strong mental model for how that would work.
I mean, it feels a little bit like you could do dimension reduction after doing a round of simulation, right? You sort of like, oftentimes these are geographic models. And so maybe you don't want a strictly geographic model, but a slightly higher dimensional model that represents something about the geography of the word structure.
I agree.
That's cool. So it's easy to go sort of off the rails here, but just sort of a speculative question. What you all are doing also is amazing, and Fiddle sounds like a great addition to thing. And sort of like, the beginnings of what is the beast for deep learning in this area? I guess, just to speculate a little bit, that's not exactly the way that things go with, say, language models, right? Like nowadays, like people are sort of like, ChatGPT or GPT is this like sort of universal thing. It's not like you train your own GPT exactly to do a task. You take the existing GPT and you might do some fine tuning or something like that.
Yeah.
So it feels a little bit like, is there some universal deep learning something that one could later fine tune for a specific task?
My opinion would be just realistically thinking about how science works and how expertise in areas comes and goes and how funding works. I think it seems difficult to sustain a massive project that handles every conceivable model and is properly debugged and maintained and so forth. That seems difficult to imagine. Even Beast, and I won't take offense, but I work on RevBase, and Beast is amazing software, and it's definitely, I would say, one of the probably the most polished piece of Bayesian software out there for the widest set of use cases. RevBase definitely does things that Beast doesn't do. And it's not because Beast couldn't do it, it's because there's not enough programmers to actually sustain that, right? So I think one thing about whatever happens, I kind of hope that with this deep learning stuff, it's almost like having a Nexus file as a common format or a Nuix string as a common format. I do think we need to have some kind of standard for how different deep learning frameworks are thinking about phylogenetic models. Because if nothing else, that lets people work on their own individual packages. Like Fiddle is just going to be one of hundreds of packages at some point, I'm sure. But it would be nice if there's a way of taking what everybody's doing and having a basis of communication. If the challenge that Ammon and I came across is that when you're learning about these new phylogenetic deep learning approaches, they're so new. Everybody's trying to figure out how to do this. And so everybody is coming up with their own custom pipeline. And they all have different assumptions. They're written in different languages. They have different tensor formats and so forth. It really limits how one person would take their findings and try to reproduce it in another framework. Right now, you basically would have to code everything from scratch again. And not only does that limit how rapidly people explore models, but I think it's going to impede the extent to which we confirm that these methods are well behaved or not, and for what situations they behave nicely, right?
Yeah, I mean, I hope we sort of have the chance to start again, right? Like, so let's all try to get behind a single framework and not have Beast 2.
Yeah.
Great. Okay. Well, I mean, I could go on, but I feel like we should end here. We're at an hour and 15 minutes.
Yeah.
I really appreciate your time.
Well, those were the nicest trolling questions I've had, so thanks.
All right. Thanks again, y'all. And yeah, the next talk is going to be about integer representations of phylogenetic trees and if we can use this for gradient-based inference from some or but. Bye.